So, cell theory states that all cells come from other cells. It's one of the foundational theories in biology. But since, like, everything eventually dies, that means that one of the fundamental tasks that cells have to perform is to reproduce themselves, to make more of themselves. Remember that reproduction was one of the qualities of life. So, basically, like, in order for things to keep on living, you have to have some way for, one cell to become two cells. And there are a number of different ways that this can happen. But it's regulated, at least in humans and other animals and most plants, by something called the cell cycle. Just like you have a life cycle, you're born as a baby, and then you grow up, and you get bigger, and you go through adolescence, and then you go through adulthood, and blah, 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 and eventually you die. But just like there's, a life cycle, there's also a life cycle of the cell, and this is called the cell cycle. So the cell cycle has several parts to it. And it goes around and around in a circle. The two main divisions are what are called interphase and mitotic phase, or M phase. The mitotic phase, or mitosis, is where the cell is actively turning into two cells. Interphase is all of the other time. Interphase is all of the time when the cell is not actively dividing into two cells. And so most cells spend most of their time in interphase. There are some very rapidly dividing cells that go through interphase very quickly and spend most of their time in mitosis. This is something that happens, say, for instance, very, very early after fertilization, when cells are dividing quite quickly. But under most circumstances, most cells spend most of their time in interphase. And interphase is divided into three subphases. They're called G1, S, and G2. The G stands for gap. One of the things that you have to do if you're going to replicate yourself is to make a copy of your DNA, right? You receive a certain amount of DNA. You need to keep that same amount of DNA. And so that means that, like, when a cell divides, it can't just let half of its DNA go to each cell without ever replicating it. So your cells can be basically in G1, and the G stands again for gap. In G1, the cell is growing. In S phase, S stands for synthesis. It's copying its DNA. So it starts off with one copy of its DNA, and then in S, it makes a second. And then you have a second gap in G2. So the DNA in eukaryotes is kept in a form called chromosomes. And these chromosomes are long strands of DNA that are packed up into these tight little bundles that look like this. And so during G1, you have, technically you have 46 chromosomes. But I'm just going to draw 3 here. And chromosomes all have a centromere, which is the thing at, well, it's not always located at the center, but it is considered the center of the chromosome. And then they have legs, usually 2 of them, one going up and one going down. When the DNA is replicated, the number of chromosomes stays the same. But instead of each chromosome having one chromatid, so this thing with a centromere and the 2 legs is a chromatid, this is a chromosome, and this is a chromatid. After S phase, the DNA is doubled, but each copy remains bound to the original at the centromere. So you see here in G1, this organism has 3 chromosomes. Each chromosome consists of 1 chromatid. During S phase, the DNA is replicated, but the number of chromosomes remains the same, 3 chromosomes. But now each chromosome is made of 2 chromatids bound together at their centromeres. This will be important in just a moment. In the cell cycle, the cell grows through G1, doubles its DNA in S, and usually continues to grow or just does its job during G2. But during G2, it's also preparing to enter into mitosis. And so mitosis is the process by which cells double. Note that it's not the process by which DNA doubles. DNA doubles during interphase, during S phase. That's when DNA replication takes place. Mitosis is when the cell replicates. And mitosis has 4 or 5 phases, depending upon which book you're looking in. I'm going to talk about 5 phases of mitosis. Those phases are prophase, prometaphase, metaphase, anaphase, and telophase. In case you were wondering, prometaphase is the one that sometimes is omitted, depending upon the textbook that you're looking at. Let's examine what's happening in the cell during each of these stages. So here we have a cell before mitosis. So here we have the nucleus, all right, and it's got a whole bunch of uncondensed chromatin. So this is a whole bunch of DNA just sort of like blah, 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 unwound, swimming around, right? It's not totally disorganized, but but is also not wound up and visible. The nucleolus is a special part of the nucleus. We have these centrosomes, right? 2 centrosomes, and each centrosome contains 2 centrioles. If you'll recall, these are non-membrane bound organelles that make microtubules, which are these like highways, these structural highways, part of the cytoskeleton that the cell uses to give itself structure and to move things around. You also have a bunch of organelles, and I didn't draw all of the organelles here, but here we have endoplasmic reticulum, here we have a Golgi, and I put a couple of mitochondria here just to illustrate a point. All right? There's more organelles than that. Those are the ones that I wanted to keep track of here. So here we have a cell. This is in interphase, right? Probably, let's say this is in G2. So prophase. In prophase, the first phase of mitosis, the chromatin, or the chromosomes, condense, which means that instead of just having their DNA, blah, 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 all strewn about, it gets packed up tight into a bunch of individual chromosomes. And here you can see these chromosomes each have 2 chromatids, right, bound together at the centromere, which is that little green thing there. In addition, the centrosomes begin to migrate to opposite sides of the cell. During prometaphase, the internal membrane-bound organelles are all disassembled. They're packed away neatly, like all of their components are packed away very, very nice, very neatly, very organized so that they can be reconstituted later. But you can see that the endoplasmic reticulum and the Golgi have both been disassembled and put away. The nucleus here has also been disassembled. I faded it so that you can see where it was. Note that the mitochondria don't. They stay. And that's because mitochondria are like a special sort of organelle that actually reproduces on its own cycle because they're descended from bacteria. So prometaphase here, we have our chromosomes condensed. They're just sort of spilled out into the cell. The organelles have dissolved and been packed away. And the centrosomes are now on opposite sides of the cell. The centrosomes make something called the spindle apparatus. That's these tubes coming off of it. These are microtubules, and they're going to attach, at least many of them are going to attach to the centromeres of these chromosomes. So they're like little ropes, like little Spider-Man ropes that shoot out and grab hold of the centromeres. Pro, by the way, means like beginning or first, right? So prophase is the first phase. Metaphase, meta means middle. And so in metaphase, what happens is these chromosomes now line up single file down the center, or what's called the midplate of the cell. So that's why this is called metaphase. It's distinguishable because you can see all the chromosomes lined up down the middle of the cell, and you've got your spindle apparatus coming out to it. In anaphase, what's going to happen is these chromosomes, before anaphase, a chromosome had two chromatids connected at the centromere. Now we're going to disconnect the centromere. And each of these two chromatid chromosomes is going to break in half into two one-chromatid chromosomes. At the same time, the spindle apparatus is going to reel them in, dragging them towards the polar opposite sides of the cell. Like that. The cell also lengthens during this phase as the two spindle apparati push against each other. So we pull the chromosomes to opposite sides. We've now doubled our number of chromosomes by splitting each one in half. The last phase is telophase. And telophase has basically two parts to it. In the first part, what's going to happen is the nucleus is going to reestablish itself. We're going to unpack and reassemble our nucleus around the chromosomes. And we get cytokinesis, where the cell will actually pinch shut. It sort of gets pulled in tight until these two things actually bubble and let go from one another, and you end up with two cells. At this point, the chromosomes within the newly formulated nuclei uncondense back into their normal state, and the organelles are remade. So you put the Golgi and the endoplasmic reticulum and everything like that back together. So this process of the cell physically dividing into two is called cytokinesis. And that is the basics of the cell cycle. You go through interphase with G1, S, G2, and then you go into M phase, which is mitosis, with its 5 phases. And at the end of that, you end up back where you started, except now you have two cells.